Chapter 3 is an introduction into prokaryotic cells. So, prokaryotic cells are classified into two different domains, archaea and bacteria. And as I mentioned in Chapter 1, we have three domains. The other domain is eukarya, but those are eukaryotes, so they actually have a nucleus. So again, key thing to know with prokaryotes, they do not have a nucleus. So we'll discuss a few of these different features of the bacterial cells or in this chapter. So first thing I'd like you to be aware of is that prokaryotes have a lot of different sizes, shapes, and arrangements. And one term to know is called pleomorphic, and that's when bacteria can take on different forms. So when we study microbes, we have to be aware that they could change shapes if they're pleomorphic or size or whatever, depending on what we're growing them in. So some of the common shapes then in bacteria would be bacilli, or rod-shaped, cocci, or spherical, or spirochetes, which are spiral-shaped. Now, Vibrio, Stella, and Coccobacilli aren't quite as common, but they are ones we would want to be aware of. Now, bacteria can be in different arrangements. When we look at the arrangements, anytime you see the term diplo, guys, diplo means paired. So there's two, cocci. Strepto means that there are chains, so strepto is chain. Staphylo means grape-like or clustered. Now you can have cocci, which are round, but you can also have bacilli, which are rod. So diplobacilli would be paired rods. Streptobacilli would be chains then of rods. Now a palisade arrangement is when you have rods that are actually in like a stair step or steps going up with the way that they're attached. So again, here's a good idea of single, diplo, strep, and staph. And then when we look at the rods, you can see a nice chain there. Palisades, they're kind of stacked more this way. And again, these are just some of the other shapes that are out there as well. Now, when you guys hit A&P and maybe in some other courses, you know that our cells, because we're eukaryotes, we undergo mitosis and meiosis. Meiosis is for gamete production. But our regular cells will divide by mitosis. Prokaryotes divide by binary fission. So please know the steps of binary fission. In the first step, the DNA is copied. Then the cell is going to grow. Then what happens is, and when we talk about bacteria in particular, normally the chromosome, there's one and it's round. So that copied chromosome then is drawn to the opposite ends of the cell. Then a little partition called a septum starts to form, and then eventually the two bacteria get walled off and they separate from one another. So this slide basically shows what the text in the last slide showed. So you have DNA being copied, the cell's going to get bigger, then eventually you see that the two chromosomes start moving. Then this septum starts to form. Eventually that's going to start to pinch off. Then at the end, the daughter cells will separate. Now, plasma membranes are boundaries that many cells actually have. Some have inner and outer, some just have an inner, etc. And in bacteria, there's also a cell wall in addition to a plasma membrane. So membranes have selective permeability. So that means that they will allow certain things to go in and out. And then some things cannot go in and out. So they select what they're going to allow in and out. Now, bacteria have peptidoglycan in their cell wall. Now, archaea is also a type of prokaryote, so please know that they use pseudomurin. It's not really peptidoglycan, so when you see pseudo, that means false. So they use what's called pseudomurin instead of peptidoglycan. Now, for your exam, you're going to have to know the differences in the cell walls of the gram-negative and gram-positive. So let's look at the gram-negative. First thing to notice is there's an outer membrane. Also notice we have lipopolysaccharides and porins. So as the semester goes on, we'll talk more about that. But that's the outer membrane. Then they have a thin peptidoglycan. And then they have the plasma membrane. If we look at the gram-positive, they have a very thick peptidoglycan. They also have teichoic acids. And there's another type of teichoic acid called lipoteichoic acid, not shown here, but that also goes all the way through. And they also have a plasma membrane. So again, with the gram-negative, thin peptidoglycan, they have an outer membrane, and in the outer membrane, we have lipopolysaccharide and porins. Gram-positive have thick peptidoglycan, teichoic and lipoteichoic acids. So when you do the gram staining, guys, gram-negative, pink, gram-positive, purple. Now, in general, gram-negative are harder to kill than gram-positive. The outer membrane is a very thick and protective barrier. So it protects it against damage from some types of degradative enzymes, drugs, detergents, and disinfectants. Now, there are the porins, and please know that the porins are protein channels that form a pore, so that allows things to go in and out of the gram-negative. So when you do gram staining, you're going to see then, as I mentioned, gram-negative have this nice pink color, and gram-positive have a nice dark purple color. Acid-fast will detect mycolic acid. Please know, for your exam, that there are two genera of bacteria that are considered acid-fast. Those are Nocardia and Mycobacterium. The genera are Nocardia and Mycobacterium. And TB and leprosy are considered Mycobacterium. Now, there's a really strange species of bacteria called Mycoplasma. Those lack a cell wall. Why would that one be important? So if you've ever heard of walking pneumonia is caused by Mycoplasma. So that micro is very easy to distinguish from other types of bacteria because it doesn't have a rigid cell wall, so it kind of blobs out a little bit. So we're going to talk a little bit about diffusion, and we can have passive and active types of transport. So diffusion itself is the passive movement of substances from high concentration to low concentration. So let's talk about a couple of types. First type would be simple. So simple diffusion is when you move molecules from a high to a low concentration, no energy involved. If you have facilitated diffusion, you're moving substances again from high to low. However, you have to get assistance with channel proteins and carrier molecules. So again, facilitated, you move high to low with the help of channel or carrier proteins. Osmosis then is the movement of water from a high to a low concentration. So we have three types of environments we could have. In hypertonic, the water moves out and the cells shrivel up. In hypotonic, water moves in. Cells are going to swell up and potentially burst. Isotonic, no net movement, so basically the cell is going to stay the same in size. So again, hyper, water moves out. Hypo, water moves in. Isotonic, no net movement of water. Now with active transport, please know that it requires energy and it requires carrier molecules. Now, here's the difference. between this and passive transports. Active transport can transport molecules from a low to a high concentration. Low to a high concentration. And again, here's an example of some different types of active transport. So, some other features of prokaryotes I'd like to cover. First of all, flagella. Flagella actually help microbes move. And what do they move in response to? Well, first of all, please know if they move in response to chemicals, we call that chemotaxis. If they move in response to light, phototaxis. And some of them move in response to oxygen. So that would be aero. So chemo, chemical, photo, light, aero, oxygen. For your exam, please know that we have different flagellar arrangements. Monotrichus would have one single flagellum. Lophotrichus has several in one spot. Amphitrichus, you're going to see flagella at each end. And for peritrichus, flagella will be all over the surface. Fimbriae are short bristle-like protein structures that stick out of the surface of a cell. It helps prokaryotes stick. So these little sticks-like structures here that are sticking out, they actually help microbes to stick. So they can stick to you and they can stick to each other. Another structure that looks similar are called pili, but notice how much longer these look. They tend to be more rigid, and they will also help things stick. However, the big important thing to know with pili is they will allow and help in gene transfer through what's called conjugation. So two bacteria come together, they can share then their genetic information. A glycocalyx is a sticky carbohydrate outer layer. It helps bacteria stick. So in order for you to get an infection class, please remember the microbe has to stick to you in order for that to occur. So there's two types of glycocalyx I'd like you to know. A slime layer, unorganized. It's a loosely associated type of glycocalyx. A capsule is well organized. It's very tight. It's smooth. If you look here, you can see how smooth that is around the bacteria. So these clear areas are the capsule. This fuzzy area here around this bacteria, and you can see it all the way around, that's a slime layer. As far as ribosomes go, all you have to know for your exam is that the prokaryotic ribosome is 70S. Prokaryotic, 70S. So that's your complete ribosome here. Please take a screenshot, okay, of this slide, upload to the Dropbox. Endospores we'll talk a lot about this semester, but please note that they're inactive structures that allow certain cells to enter a dormant state. These will form when bacteria are stressed. So, any of you have ever had a patient with C. diff? C. diff is a spore-forming bacteria. Anthrax is a spore-forming bacteria. So if you stress those cells out, they just turn into these endospores, and then later on when you take away the stress, whether it's a treatment, whether it's heat, whatever, they can start to grow again. So again, these are just a few examples of microbes that produce endospores, and we'll talk about them later on this semester.